All right. Hey, everyone. You've made it to our last lecture of the semester. And today is all about the ear and the eye. First, we'll go over the anatomy of the eye, the cellular makeup of the retina, and talk about how light passes through the eye. Here's a look at the external anatomy of the eye. The cornea is the clear layer that covers the eye. The eyelids are going to be called your palpebra. And the palpebral conjunctiva is a lining on the inside of the eyelid. There's also a bulbar conjunctiva, which surrounds the outer border of the cornea. When the conjunctiva get infected, they develop conjunctivitis. That's what we would know as pink eye. Then we have the tarsal plate, which is going to hold our tarsal glands that secrete an oily substance to lubricate the eye. And the eyelashes work to prevent dust and debris from getting into the eye. Here's another view of the exterior of the eye. Specifically, here we are looking at our lacrimal areas. The lacrimal glands secrete lacrimal fluid. The lacrimal fluid will flow across the eye into the lacrimal caniculus and then the lacrimal sac, and eventually down the nasal lacrimal duct and into the nose. The lacrimal caruncle is where the sebaceous glands are going to be located. And then lastly, we have the lateral and the medial commissures. And that's where the eyelids join together on either side. And this is a look at the 6 muscles in the eye. There are 4 rectus muscles so that they're straight, and there are 2 oblique muscles, so they're going to be angled. They all act in accordance to their name. So for instance, the lateral rectus moves the eye out laterally. Elevation of the eye is going to mean to look up, and depression means look down. And this is also where we'll start connecting the cranial nerves to other structures in the body. So if the lateral rectus is going to move the eye out laterally, then the muscle is going to be innervated by the abducens nerve, since that nerve works to abduct the eye. So study this chart and the actions of each muscle and connect it to the cranial nerves. Now we're looking at some of the internal anatomy of the eye. The eye is divided into 2 segments. We have the anterior and posterior segments, and the 2 segments are separated by the lens of the eye. The anterior segment is filled with a fluid substance called the aqueous humor that is constantly being produced and filtered your entire life. And the posterior segment, it is filled with a thick gel-like substance called the vitreous humor, and that remains the same your entire life, and it's not constantly being produced. Within that posterior segment, the retina in yellow detects light from the environment, and deep to the retina is the choroid layer that picks up extra light that may have gotten through the retina. Surrounding the posterior segment is the sclera, which makes up the white of your eye. The lens is held in place by the ciliary zonules attached to the ciliary processes, and the ciliary processes are extensions off of the ciliary muscle. The ciliary muscle and ciliary processes together are referred to as the ciliary body. And then lastly, the ora serrata separates the ciliary body and the retina. The iris is the colored portion of the eye. The pupil is the very center of your interior portion and will dilate or constrict depending on the amount of light that is available. The optic nerve comes into the posterior portion of the eye and innervated at the optic disc. This is also called your blind spot. Above the blind spot is the macula luteae. The macula luteae is a region of the retina with a very high density of rods and cones. Rods and cones are photoreceptors that help us detect light and color. Cones let us see color, and rods let us see black and white. Within the macula luteae is the foveus neutralis, which is a region that it only contains cones. This figure is a view of the ciliary body. You can see the ciliary muscle and the ciliary processes and these ciliary zonules. And in the middle, you see the lens. You can also see the ora serrata is the junction point where the ciliary body meets the retina. This is a microscopic view of the retina. Present here are rare bipolar neurons. These neurons innervate our rods and our cones and let us see light and color. At the very posterior portion, we have our pigmented layer of the retina. And so the left side of the slide is going to be the anterior portion, and the right side of the slide is going to be the posterior portion of the eye. Light is going to come in on the anterior portion, passing through to the posterior portion where the rods and cones are. And then the visual information is then processed, and the signal output is going to move from the posterior side to the anterior side. And this is a histology slide of the retina. You have the choroid layer, then the pigmented layer of the retina. Then we have our rods and cones and our bipolar neurons. And finally, we have our ganglion cells. Here is a dissected cow eye, and it's very similar to the human eye. At the top, you'll see a lot of adipose tissue surrounding the eye that acts as a cushioning for the eye. Then we can see the cornea, which is dark because of the preservatives that are typically used, cause the discoloration of the cornea. Typically, the cornea is clear. Then we have the sclera and the optic nerve. And then on the outside of the eye, or on the inside of the eye, we can see the ciliary body, the lens, and the optic disc. So now we'll start looking at some of the terms in regards to vision and certain visual tests that you'll need to know. So light passes through the anterior segment and through the lens to the posterior segment. The lens is going to bend the light rays and cause the image that you see to be flipped upside down. This bending of the light is called refraction. That image is then processed, and this thing is sent back towards the anterior portion of the eye and flipped back to the upright position. If your eye is shaped differently, then the refraction of the image will be inaccurate, and you need to get corrective lenses to fix the problem. Once light passes through the lens and bends, the light will converge into a point and hit a focal point on the posterior side of the eye. If the light hits the focal point correctly in the right place, this is called an emmetropic eye, and corrective lenses are not needed. If the eyeball is too long, when light passes through the lens, the light will converge too soon and won't hit the focal point correctly. This is called a myopic eye and is known as being nearsighted. If this is the case, you will have difficulty seeing things that are far away. To fix this problem, you will need lenses with a concave shape to make the light converge at the correct place. If your eye is too short, then you will have a hyperopic eye, and the light will not converge soon enough to hit the focal point. This is also what you would call being farsighted, and to fix this, you would need a corrective lens with a convex shape. So remember, concave shape for a myopic eye if you're nearsighted, and a convex shape for the hyperopic eye if you're farsighted. And then this is a test to see if someone has an astigmatism. And astigmatism is a condition where the eye is not completely round. If you do not have astigmatism, you should be able to look at this chart and see all the black lines very straight and very clear with no blurriness. If you do have an astigmatism, then you would see some of the lines appearing blurry. This is an image of a retina that you would have taken if you go see your eye doctor. It is used to check if the retina is healthy and if everything is attached correctly. You can see the optic disc, and that's where all the vasculature of the eye will branch out from. And then you can also see the macula lutea, where a lot of cones are going to be concentrated. And then these are the visual conditions you're going to need to know. So emmetropia is a normal vision, and it means the eyes are the correct shape and do not need corrective lenses. Hyperopia is farsightedness, and you have difficulty seeing things close up, and you will need a convex shaped corrective lens. Myopia is nearsightedness, and you will need a concave shaped corrective lens. And this is because you have difficulty seeing things that are far away. Presbyopia is old vision and is the gradual loss of your eye's ability to focus on nearby objects as you age. And it's not the same thing as being farsighted. This is why like our grandparents have really hard time seeing things up close, like reading on their phone or reading the newspaper. And then lastly, we have our astigmatism. That's blurriness due to an uneven curvature of the lens or cornea, meaning the eye is not completely rounded. So next, we'll go over the anatomy of the ear. The ear is divided into three different parts. You have the external ear, the middle ear, and the internal ear, also called the labyrinth. The auricle is the top of your external ear and is made of elastic cartilage. The middle is called the helix, and then the bottom is your lobule or your earlobe. The external acoustic meatus connects the external to the internal environment. In the middle ear, we see our tympanic membrane, which is also called the eardrum. Also in the middle ear, we have three tiny bones called the auditory ossicles. First, we have the malleus or the hammer. Then we have the incus or the anvil. And lastly, we have the stapes or the stirrup. In the inner ear, we have everything else. We have the cochlear and vestibular nerve innervating here. And we have the pharyngotomatic tube or the auditory tube connecting the pharynx to the inner ear. And here's a look at the inner ear. There is the cochlear portion responsible for hearing and the vestibular portion responsible for helping maintain balance and equilibrium. The vestibular portion is going to contain the utricle and saccule and vestibule and the semicircular ducts and canals. At the base of the anterior, posterior, and lateral portion of these semicircular ducts is the cristae ampullaris and is innervated by the vestibular nerve. In the cochlear portion, there is the cochlear duct inside the cochlea. The cochlear duct makes the spiral shape that gives it a snail shell shape. And in the inside of the cochlear ducts is the spiral organ. So this is an image of the cristae ampullaris. The cristae ampullaris is held by the cristae ampullaris. The crista ampullaris is composed of hair cells and supporting cells. Below the crista ampullaris is the vestibular nerve, and above the crista ampullaris is the hair bundle. So when you spin in a circle, the endolymph flows backwards and pushes the hair bundle in the opposite direction. And when you stop spinning, the endolymph continues to flow backwards, pushing on the hair bundle. And this is what makes us feel dizzy, like the room is still spinning, even though we have stopped spinning. The left side gives a look at the inside of the cochlea. Inside the cochlea is the cochlear duct, also called the scala media, that contains a fluid called endolymph. Above and below the scala media is the scala vestibuli and the scala tympani, that both contain perilymph. In the middle of the cochlear duct is the spiral organ. The spiral organ has the basilar membrane where the cochlear nerve will innervate. And then above the basilar membrane are supporting cells, and these supporting cells will come into contact with the tectoral membrane. And here is a histology slide of the inside of the cochlea. The hair receptor cells and the supportive cells support the tectoral membrane, and the basilar membrane is below that. The scala vestibuli and the scala tympani are going to be holding that perilymph. So this figure shows how we hear sound waves. Higher pitched sound waves will produce a greater amount of hertz, and a lower pitched sound wave will produce a lower hertz measurement. Depending on the portion of the basilar membrane, the length of the basilar fibers will differ, some being longer or shorter. And the different fibers will detect sound waves at different hertz levels, and how the sound waves hit the fibers in each section will determine what you hear. Just know that that's how it's done. Don't worry about too much about these specific numbers and hertz ranges. Okay. There are two hearing tests we'll talk about, the Weber test and the Ryan test. The Weber test distinguishes possible conduction or sensory deafness. Conduction deafness is indicated by hearing a louder tone in the affected ear. Sensorineural deafness is indicated by the tone being heard in the unaffected ear. A normal test will have the tones sounding the same in both ears. So a tuning fork is tapped and then placed at the top of the forehead, and the sound appears to be louder in one ear than there is possible deafness. The Ryan test will assess only conduction deafness. A strut tuning fork is placed on the mastoid process behind the ear. A normal test result occurs when the individual hears the tuning fork by air conduction following bone conduction. And then a conduction deafness result occurs when the individual hears a tone by bone conduction following air conduction. So this is an example of what the Weber and the Ryan test will look like. All right, so that's it for this semester, guys. Go over this a couple times. We have practical coming up soon, so good luck, and I'll see y'all in lab.